Assalamualaikum warahmatullahi wabarakatuh. We are from Group 11 and today we are excited to share our presentation for Food Biotechnology FST 23602. Our topic is on kimchi as a fermented food. Let me introduce our team members: Imam, Zulhisham, Zahra, Sofia and me, Shira. Moving on to introduction. Kimchi is a famous Korean side dish or banchan made from fermented vegetables and spices. It is a staple of Korean cuisine and a classic example of lactic acid fermentation. The core ingredients usually include napa cabbage or Korean radish, seasoned with a blend of chili powder, garlic, ginger, and fish sauce. This creates a complex flavor profile that is sour, spicy, savory, and often has a slight natural feel. How does it become kimchi? It undergoes a spontaneous fermentation process where beneficial bacteria naturally present on the vegetables thrive. The transformation that no starter culture is needed. Set plays a critical role by drawing out water and inhibiting spoilage bacteria while allowing healthy, salt-tolerant bacteria to thrive. Kimchi is globally recognized today as a probiotic powerhouse rich in vitamin A, B, and C. Beyond nutrition, it is a significant cultural heritage. In fact, the traditional practice of making and sharing kimchi, known as kimjang, is officially recognized by UNESCO.
Assalamualaikum, my name is Siti Zahra Tujanna binti Muhammad Karudin, metric number 083173. On this slide, I will explain the characteristic of bacteria involved in kimchi fermentation. The common bacteria found in kimchi are Leuconostoid mesenteroids, Lactobacillus plantarum, and Bacillus corensis. First, these bacteria produce lactic acid by converting the natural sugar in vegetable during fermentation. This process gives kimchi its characteristic sour taste and create a favorable environment and beneficial bacteria. Second, they are salt tolerant. Since kimchi is prepared using a breeding process, these bacteria can survive and grow in salty conditions where many harmful microorganisms cannot. Third, they can grow under anaerobic conditions, meaning they require little or no oxygen. This is important because kimchi is usually fermented in tightly closed containers. In addition, the lactic acid produced help preserve the food by lowering the pH and inhibit the growth of spoilage and pathogen microorganisms. And finally, these bacteria contribute to the unique flavor, aroma, and texture of kimchi by producing organic acid and other fermentation compounds. Lastly, these characteristics make lactic acid bacteria essential for successful kimchi fermentation and quality.
Moving on to the authentic traditional manufacturing method, the process begins with phase 1, that was brining. The napa cabbage is halved and coarse salt is applied manually to every single leaf layer. The critical osmosis process takes 12 hours, followed by 4 hours of draining to completely remove excess moisture. The defining features of this traditional method lies in phase 2, the seasoning test. Instead of plain water, it used a rich dashi stock called yuksu, made from anchovies and kelp. The stock is combined with gochugaru, rice paste, greens, and blended oyster, which act as a natural biotechnological booster to stimulate beneficial lactic acid bacteria. Finally, in phase 3, the paste is coated roughly between every leaf layer. The cabbage is rolled tightly and wrapped with its outer leaf to prevent oxidation, then packed into an airtight container for a slow, mature anaerobic fermentation lasting 1 full month. Thank you.
Now, we move to mechanisms that occur during the process, which is kimchi fermentation. First is a lactic acid fermentation, the main fermentation process in kimchi. Lactic acid bacteria, LAB, such as Leuconostoc, Lactobacillus, and Bacillus, convert the sugars in vegetable into lactic acid. This process lowers the pH, produce a sour taste, inhibit the growth of harmful microorganisms, and extend shelf life. The second mechanism is heterolactic fermentation. Some LAB produce not only lactic acid but also carbon dioxide, ethanol, and other flavor compounds. This contribute to the distinctive aroma and slight gas formation in kimchi. The third mechanism is alcoholic fermentation. Yeast naturally present in vegetable can convert sugars into ethanol and carbon dioxide. especially during prolonged fermentation. The alcohol content is usually very low. Next, we have acetic acid fermentation. If oxygen is present, acetic acid bacteria can convert ethanol into acetic acid, increasing the sour taste and changing the flavor profile of kimchi. Other than that, we have carbon dioxide production. CO2 is produced during fermentation, causing the formation of bubble and increase in pressure in closed container. As for the last mechanism, we have protein and amino acid decomposition, which is enzyme and microorganism break down protein into peptides and amino acid, which enhance the umami flavor of kimchi.
To summarize, kimchi is a very nutritious and culturally significant fermented cuisine that combines seasoning, veggies, and the action of good lactic acid bacteria to produce a product full of probiotic, flavor, and health benefit. In addition to extending its shelf life, the fermentation process improves its functional qualities, which include enhancing digestion, increasing immunity, and promoting gut health. So kimchi maintains its safety and quality while developing its distinct sour, spicy, and umami taste through carefully regulated lactic acid fermentation. All things considered, kimchi is a great illustration of how traditional fermentation can turn basic ingredient into a tasty, healthy, and microbiologically safe dish that is still prized in Korean cuisine and around the world.